Hello, today we are going to be talking about the metabolism breakdown. Okay, so first things first is what is metabolism, okay, and why is it important to you? In the world, everything is going to require energy, whether it's you running, you sleeping, you resting, anything that all that you're doing. If you're taking a selfie, it requires energy. Okay, this is a good example of a hummingbird that's using its energy for its prolonged periods of flights. As you all know, hummingbirds flap their wings numerous times within a minute. So they actually do need to process that food that they're intaking and let it undergo chemical reactions to thus give them the energy they need for flight. Bioenergetics is the term used to describe the concept of energy flow through living systems such as cells. Let's get this way. Okay, so most life forms on Earth get their energy from the sun. That's the original place where we're going to be getting it from. And here's a nice little food chain that we can follow to understand how this gets transferred from one form to another. For example, here the sun is actually allowing the producers or the autotrophs, the plants, to create energy by using its sunlight energy. Then we have primary consumer, right here the grasshopper, who's going to eat the grass. Then we have a secondary consumer, the frog's going to eat the grasshopper, and so forth, until finally we get to the final consumer, which is this hawk. And this energy that was originally created from the autotrophs or the producers gets transferred from one form to another. Okay. Energy in the world of life. So sustaining life's organization requires ongoing energy inputs. And what is energy? We're talking about energy, but energy is actually the capacity to do work. Okay, that's why we've talked about kinetic energy, and we've also talked about potential energy. Okay, throughout this that food web we just looked at, one form of energy was converted to another. So some examples of some kinetic energy, which is the energy that's involved with motion or objects in motion, are light, heat, electricity. Okay, and some energy in chemical bonds are known as potential energy. Remember, potential energy is stored energy. It has a potential to do the actual work, but it's not in the motion of doing so. So a good example would be imagining those ping pong machines. When you pull the spring back, not releasing that lever, but if you hold the lever back in a ping pong machine, okay, that's actually going to allow you to maintain that potential energy, that stored energy. Okay. Also, if you just think about it like a spring on the doors, you guys, some of us have the springs that actually hold the doors from getting from ruining the walls. If you push that spring together and maintain it and just hold it there, okay, that has potential energy now because the energy is stored. If you release it, then that's actually the stored energy that you're allowing to be released. So for this lecture, we're going to be talking about that energy that's stored in chemical bonds. Okay, cellular processes such as the building and breaking down of complex molecules occur through chemical reactions. Okay, there's it could either be spontaneous and release energy. That would be an exergonic reaction. Exer, EX, meaning that it's exiting. It has a release of energy. Okay, and then there's also endergonic reactions, which is going to be the building up of a molecule that does require an input of energy. So there's two different kinds. You can either build a complex up or you can break it down. Okay. What is metabolism? Okay, I know a lot of people think right away of, oh, mine's fast or oh, mine's slow. But what are we talking about when we talk about metabolism? When we talk about your metabolism, we're talking about all those chemical reactions that occur inside of the cells in your body that are used to actually consume those glucose molecules that you're intaking and generate energy ultimately. So it's basically how fast is your body doing it? How fast is your body undergoing those chemical reactions to release the energy that you need? And why do cells need energy? Well, cells need energy to constantly replicate, create proteins where it's needed, and actually to repair some damaged tissue as well. So lots and lots of uses for cells to actually use the energy that it requires. Okay. So energy in the world. Okay, this goes back to physics and learning about thermodynamics. So the first law of thermodynamics is energy is never ever created nor is it destroyed. It's just transferred from one form to another. Like in the original picture we saw earlier, where we had the actual sunlight energy and it going into the actual plants or the autotrophs, and then harnessing the energy and creating glucose molecules out of it, which is actually endergonic reaction because it's creating a larger molecule. And then there's also the second law of thermodynamics. So the second law of thermodynamics is saying that entropy, which is a measure of the dispersal of energy in a system, will always increase spontaneously. Okay, what is entropy? It is actually interpreted as the degree of disorder or chaos or randomness in a system. And whenever you actually combine two atoms and create a bond between them or build a larger molecule up, which is anabolism or a endergonic reaction, okay? Whenever you do that, it actually will decrease the amount of chaos in a system or entropy in a system, okay? Also, every time that energy is transferred, there is a bit of heat energy that's lost with that transfer. And energy, like we said, it's never created nor destroyed. It just gets transferred from one form to another. So here's a view of it from your book that shows this and shows what's happening, kind of like the food web we just looked at. So energy for life on Earth comes from the sun. So energy comes in from the sunlight, okay? Producers or autotrophs are able to convert that energy into glucose or other forms of energy required by consumers. So energy flows through producers, then consumers. Energy is transferred many times back and forth. But once again, like the law of thermodynamics, the second law, it does say with each transfer, some energy escapes. So there will always be a loss of energy, and that escapes usually as heat energy, which is unusable energy. All of the energy is eventually permanently dispersed throughout the Earth. So metabolic pathways that you all will be familiar with are going to be photosynthesis is going to be number one. This is a metabolic pathway because plants are actually using the energy from the sunlight to convert it to glucose right here for consumers to eat. Okay, so photosynthesis are when plants are actually taking in carbon dioxide, some water, and then using the light from the sun. The light from the sun hits the chlorophyll or the thylakoid membrane on a plant, and then that converts the carbon dioxide and water into this much larger molecule of glucose. Okay, and the result, which is these products, glucose, oxygen, and water, these are what we use as consumers to fuel our bodies. This is known as an anabolic reaction because we're building up the molecule. Okay, we're building up from these two tiny ones, kind of like monomers, and building this larger molecule. So it's a build-up reaction. You have to build a molecule up. That's an anabolic reaction, kind of like anabolic steroids. So after plants create those glucose molecules, we actually break those glucose molecules back down to use the stored energy inside of them. So the energy storage molecule, which is glucose, C6H12O6, is consumed and broken down by us. So the opposite of anabolic reaction is going to be catabolic reactions. Okay, so you have a much larger molecule, glucose, and then we intake our oxygen, and then it goes through the chemical reaction and releases our waste products, which is carbon dioxide, some water, and our energy that our cells require for us to maintain life. So this is the big, beautiful circle of life. The sun comes and releases its energy, sunlight energy. It goes onto the plants who undergo photosynthesis. Then they release glucose as their waste product and oxygen. Well, guess what? That's what humans run off of, sugar and oxygen. So then we intake our glucose and we undergo and use that glucose for cellular respiration. Once our chemical reaction occurs with cellular respiration, it does release, like I said, these two waste products, which is carbon dioxide, what we blow out every day, some water, And this is the most important one, ATP, which is energy for your cells to be fueled on. And then that waste product gets used back up in the photosynthesis in plants, and it just goes round and round and round. This is why a lot of people are so worried about all the destruction that we're doing to large masses of land, because we're removing all of these natural plants and trees that actually are doing photosynthesis and releasing this glucose, which is a requirement that we need to survive. So, energy in the molecules of life. Okay, first and foremost, when we're looking at a reaction, we have number one, a chemical change that occurs when atoms, ions, or molecules interact. They can either be building up or they can be breaking each other down. That's what a reaction is. The reactants are always on the left-hand side. It's kind of like what you begin with, the items that you begin with. So the reactant are atoms, ions, or it could be molecules that enter a reaction. It's how you start the entire reaction. So if we're looking at photosynthesis, okay, our reactants are going to be those two atoms that were on the left-hand side, which was our carbon dioxide and our water molecules, okay? Then the product are the atoms, ions, or molecules that remain at the end of a reaction. So our product at the end of photosynthesis are going to be our glucose molecules, our ATP, and our water, okay? So your reactants are what you start with, and here are your products, or what you end up with. So all cells store and retrieve energy in chemical bonds, okay? All cells do this. By comparing the bond energies of reactants with those of products, so basically they're saying by comparing what you started off with and what you end up with, that can tell you whether or not you had a release of energy or if you actually uptook energy, okay? And the difference is, or the differences are going to be whether it's an endergonic, which means that you had an increased energy, or an exergonic reaction, which means you actually released energy, okay? So energy in, energy out is kind of like the sum of it. Cells, like we just mentioned, are going to store energy by running endergonic reactions or building them up. So that means if you have an add of energy, then your reactants are going to start down here, and then the products which you end up with should end up higher level of energy than what you started off with, because you're going to have an input of energy, which is shown right here. So we actually have small molecules like carbon dioxide and water, energy in, and then it's going to release larger molecules, because you should have a higher amount of energy at the end of the reaction. Cells that release stored energy, so basically how we break things down and it's going to release the energy, that's going to be kind of like our cellular respiration example. Right here we're going to start off with, let's say, glucose, and then we actually release the ATP because our cells are going to use it. And then so at the very end, we are actually left with these smaller molecules such as carbon dioxide and water as our waste product. So this is a good example of this right here of an exergonic reaction would be, like I said, cellular respiration. And a good example of an endergonic reaction would be photosynthesis. So energy in the molecules of life. Right here, we actually have it where they're showing us free energy, the amount of energy at the end of a reaction, and what it looks like, or how big. This is actually showing you how big the molecules grew, or actually how much they got condensed. So kind of just like what we exactly what we just said. Right here, you're going to have carbon dioxide and water and energy input. So this is kind of photosynthesis. At the very end, look how big this actual product is. Definitely we built up, which means there was an input of energy required. So we built it up. So it's going to be an anabolic reaction. And you can also say that this is endergonic. So it's anabolism and endergonic reactions. Okay. On the opposite side over here, though, we actually go from glucose, which is a large molecule, and oxygen. And what happens is it gets broken down into these smaller ones. Remember, breaking a molecule down into smaller parts is called catabolism. So this could be referred to as a catabolic reaction, or also an exergonic reaction. Exer, EX, remember, exit. You have an exit of energy right over here. So all these chemical reactions that are occurring and we need to know what's helping it or what's not helping it occur. And you all have heard of enzymes already, okay? So enzymes are a protein that allows for reactions to occur at a greater rate than they would normally occur without the enzyme. So enzymes just help it happen quicker. It does not mean it's going to be a difference in the result or difference in the product. Exact same products, just happens a lot quicker. So this is how enzymes work. You have a substrate. In this case, it'd be this green marking right here. That's a specific reactant acted upon by an enzyme. So here's our reactant, and here's our enzyme. Our reactant, or the beginning of our chemical reaction, is going to come and it's going to attach to the active site of an enzyme. And once it attaches to the active site of an enzyme, it's considered an enzyme substrate complex. It undergoes its chemical reaction, and boom, we are left with two new products compared to what we started with in the substrate. So once again, this would be like our reactant. These are our products right here. Same exact chemical reaction would occur if this enzyme, this darker gray, that's our enzyme. If that enzyme was not there, this would still occur, but just not as quickly. So here's another good view. This is actually from your book. Here's your enzyme. Here's your substrates attaching to the enzyme. It undergoes the chemical reaction, and after it goes the chemical reaction, it is actually allowed to release the new product right here. Okay, so an enzyme acts only on certain molecules that fit its active site. So it can't act on every single substrate that's just floating around in your cell. It has to be specific. So enzymes are very, very substrate specific to what they're going to actually help break down. Enzymes, okay, we used to think that it was a lock and key model where the substrate and the enzyme were just like a lock and key, just like how you put your key in the door. It just fit in and then it changed and then released a product. Okay, that's what we used to think happened with these guys. Now they realize that it's not actually a lock and key model, it's more like an induced fit model. What that means is that the substrate comes in and attaches to the enzyme's active site, and then a conformational change has to occur. So it kind of changes its conformation around that substrate, and then at that point it can actually undergo its chemical reaction. So it needs to actually have some sort of conformational change before it can break down that actual substrate, and that's why it's called an induced fit model now. So what are enzymes doing? I know we talked about how it can actually speed up a reaction that's going to naturally occur, but how is it able to do that? So what's actually happening is enzymes are lowering the activation energy required for an actual chemical reaction to occur. So right here in this graph, you can see this yellow line. This is the actual chemical reaction occurring without an enzyme. So here's my reactants. Okay, let's just pretend like, let's say glucose is over here. It's going to undergo right here and right here, this whole line going up and down. This is going to be the activation energy. So all this is the activation energy required for this reaction to occur. It's kind of like pushing a boulder up a hill. You're going to have to push, and you have to get all the way up to the top of the hill. That's your activation energy before you can release the boulder and then allow for the chemical reaction to occur. But enzymes reduce that activation energy required for that reaction to occur. So right here in this white line, you can actually see it's the same exact reactant. So here's glucose, and then it's going to have the same exact products, but we don't have to push the boulder up as high on this huge steep hill. We can actually only push it up to here. That's what enzymes are doing. Enzymes are allowing us to reduce the amount of energy required to begin the actual transition state and result in our products. So here's another really good example or view of it. The purple line right here is an actual reactant and a product, and this is the actual chemical reaction occurring without an enzyme. But if you add that enzyme in, here you go right here, the activation energy required from this point to this point right here is a lot less than the original required for this to actually occur. Another key point here, your reactant energy are a lot lower than your product's energy. What does that tell you? Well, that actually is telling us that there has to have been an input of energy in here because our energy actually increased. versus our products and our reactants when you're comparing these two. Remember, just like how we looked at if we build up carbon dioxide and water and then we created glucose, that's what happened. It's actually increasing energy. So there has to be an input of energy, which means thus this is an endergonic reaction or an anabolic reaction. So this is really, really cool. If you always, a lot of people think of enzymes as, oh, they're only going to allow reactions to occur a lot quicker, but there are certain species of animals out there that actually have enzyme-based venom. And one good example would be knowing about brown recluse spiders. Okay, really, really interesting. Very dangerous though. This is a picture of a brown recluse spider. It actually contains the enzyme sphingomyelinase D. What does that cause? It actually causes necrosis, which is tissue death. So if you get bit by this, what it's doing are those enzymes are actually allowing your skin cells to sit there and attach to that enzyme, and then it destroys your cell membranes eventually. So not the best situation you want to happen because of it actually destroying your cell membrane. And here is actually a big gaping hole in a woman's leg that actually occurred from this brown recluse spider bite. Okay, so this is another picture right here, and this is the information right here about this woman who actually had this bite from this brown recluse spider. The victim saw a spider on her foot as she got out of the shower. She eventually had to have her whole leg amputated below the knee. Bathtubs and showers are common locations for these brown recluse spiders because they get trapped within the smooth surfaces. So it can start off as just a tiny little bite, but those enzymes are attaching to the actual tissues in your body, which is breaking them down and ultimately causing necrosis or tissue death here. So how can we regulate enzyme activity? Okay, can we increase it? Can we decrease it? How fast it happens? Yes, you can. The way you can do this is, number one, you can actually control the temperature at which the reaction is occurring. So if you increase the temperature, you're going to increase the chemical reaction timing. You can also change the pH levels, the salinity levels, and you can add or remove cofactors and inhibitors. These are all good ways to increase or decrease enzyme activity. So let's go ahead and look at the first three that we talked about. If we increase temperature of a reaction, okay, it's going to increase the reaction rate. So right here, for example, this temperature one right here, we have E. coli, okay, polymerase, which is an enzyme. If it ends in ASC, it is an enzyme, okay. So we have E. coli polymerase. What this is showing me is that at temperatures 20, the enzyme is helping the reaction occur, but not as fast as it potentially could. Then as we get higher and higher up, maybe to around 40 degrees Celsius, the enzyme is at its optimal temperature range. So now it can actually be really, really quick on helping those chemical reactions occur a lot faster. But the thing is, if you get too high of temperature, like right here, for example, if we went to, let's say 80 degrees or 70 degrees Celsius, this would actually denature that protein structure. Remember, it's a 3D or the three-dimensional structure of protein that's getting denatured, and it's basically getting deformed. So at certain temperature though, you are going to be denaturing those enzymes. They're not going to be used anymore because they will not be able to go back to its original form. Okay, same thing goes with this one right here. This is just a different kind of enzyme right here, but it's the same exact thing where you actually have right here optimal temperature range for this enzyme. So if you start at 20, it's going to be helping, but it's not going to be helping a whole lot. But if you get to the optimal temperature range of this particular enzyme, which is around 70, then guess what? You're going to be going through those chemical reactions so fast, but you will denature it if you get over 100 degrees Celsius. Okay, enzymes also have optimal pH range as well. Most enzymes in the human body have the optimum pH range of around 6 to 8. Okay, but let's go ahead and look at these. Same thing as temperature. They have a specific pH level at which they are reacting their best, or they're helping with chemical reactions the best that they can at an optimal level. So right here with pepsin, this optimal pH for pepsin is very acidic. It's around 1 maybe 1.5 pH level. But then trypsin over here, its optimal pH range is around an 8. Once again, you get too high, you're going to denature this pepsin enzyme, or you're going to denature this trypsin enzyme if you get too high of a pH. Okay. Also, salt levels affect the hydrogen bonds that hold enzymes in that three-dimensional shape of theirs. So if those hydrogen bonds are affected, guess what? We can't keep our three-dimensional shape, which does not allow the enzyme to run or work efficiently. Enzyme regulatory molecules. These are what we were talking about a little while ago. Okay. So how can we increase and decrease the rate that an enzyme acts? Okay. Well, we did talk already about the pH, the temperature, and the salinity. Now we're going to be talking about those other items, which, for example, would be inhibitors. They're going to hinder the action of an enzyme. So what actually happens is you can have competitive inhibition or non-competitive inhibition. Right here, here's my enzyme, okay? What should happen is this substrate should be coming here to the active site, joining with the substrate enzyme complex, undergoing its chemical reaction and then resulting in a new product. But unfortunately, we actually have this inhibitor, which is a competitive inhibitor, that's going to bind and take the place of the substrate. So then therefore, the substrate cannot bind anymore over here and result in the product that it should. And this can be only temporary or it could be permanent. It is called a competitive inhibitor because of the fact that it is actually fighting with this substrate molecule to get to that place first. So they're going for the same spot. Okay. Opposite though, which is competitive, this is competitive inhibition, is we have non-competitive inhibition. Okay. What's happening here is that the actual inhibitor molecule binds to an enzyme location other than the active site. So instead of this guy's substrate trying to sit here and fight with the actual inhibitor, same spot in the active site, the inhibitor actually comes around the other side of the enzyme and attaches right here, non-competing or non-competitively trying to fight for that spot. So now it's going to come over here and join, and this is called the allosteric site. When it joins over here, it still changes the conformation or the shape of the enzyme, therefore not allowing the substrate to bind to it. So it's still an inhibitor because it's inhibiting this substrate from joining with this enzyme, but it's not competitively fighting for this active site. It's going to join at the allosteric site. Okay, so this is called allosteric inhibition. It's non-competitive. Then you also have allosteric activation. What's happening here is the inhibitor directly causes the shape change in an enzyme. Okay, the changes active site to better fit the substrate. So instead of it joining right here and then causing this conformational change in the enzyme so the substrate can't bind, it does the opposite. It's going to sit here and join right here at the allosteric site of the enzyme, but that just makes this a better fit for the substrate to actually sit there and bind a lot quicker. So it can actually have allosteric activation, which just allows for better fit of the substrate itself. Now, some products itself act as a regulator later. So for example, right here, what we would have is, this is the beginning, the enzyme is right here, that's grayed out item. This is the enzyme, okay? And normally what would happen is this substrate comes, joins with this enzyme, it's undergoing its conformational change, okay? Then it becomes this first product. Then that first product will join with another enzyme that's specific for it. Then that will result in another product. Then this other product will join with another enzyme, undergo its chemical change, and ultimately end up with this end product over here. Okay, it doesn't have to go through all these steps. It can just go through one intermediate or so forth and so forth, but this is just a really good example of showing you what's going on. This last end product though could potentially come back around to the beginning of where this initial process occurred, and now it's going to attach at the allosteric site in this example of this enzyme, therefore causing non-competitive inhibition to occur. So now it's not going to let that original substrate bind anymore because now it changed the enzyme's conformation. So this triangle cannot fit in here anymore like it once did. This is actually called feedback inhibition, when the product is actually decreasing or stopping the activity of an enzyme. So one cofactor to know about would be NAD+. Okay, it's actually a coenzyme derived from vitamin B3. NAD+ can accept electrons and hydrogen ions, like you can see right here at the top. NAD+ is going to accept electrons and hydrogen ions and therefore become NADH. Okay, NADH can thus actually be broken down again and later on become NAD+, the electrons and free hydrogen ions again. Okay, so when it's actually building up and accepting electrons, okay, it is actually considered reduced to NADH. This, when it's actually going from NADH to this NAD+, that's saying that it's actually been oxidized. Okay, one good way to memorize this is by the good old saying, Leo the lion goes grr. Leo the lion goes grr. Like he roars, okay? Leo stands for a loss. If you have a loss of electrons, that means it's been oxidized, okay? If you have a gain of electrons, then it's considered reduced. So right here, like we said, NAD+ gains electrons, therefore NADH is considered being reduced. Okay, if you have NADH and you have a loss of electrons, then it's considered to be oxidized. So right here, we actually have the electrons here. We lose the electrons right here. It's going bye-bye. It's no longer with NAD+. Now, since we lost electrons, it's now considered oxidized. So right here are a couple of other cofactors you might want to consider looking at and familiarizing yourself with. We have ascorbic acid, ATP, which is adenosine triphosphate. You definitely want to know about that guy. NAD and NAD+, NADP and NADPH. These are all enzymes we are going to be visiting when we talk about cellular respiration though. A cofactor, which is you all will know about, is ATP. Okay, it is a special coenzyme. Okay, ATP, which stands for adenosine triphosphate, is a nucleotide with 3 phosphate groups. So right here we have our ribose and our 3 phosphate groups, and here's our adenine also attached to it. So that's why it's called adenine tri, meaning 3, and phosphate. So adenine triphosphate. Okay. Energy in ATP drives many endergonic reactions, okay? And what does phosphorylation mean? That means that when ATP is actually broken down, like we just talked about loss of electrons, okay? So if it's loss of electrons, Leo, remember it's been oxidized. So when ATP loses one of those phosphate groups, okay, right here goes ATP, energy is lost. ADP becomes adenosine di, which means 2, diphosphate, and here's your free phosphate over here that gets removed. When that happens, okay, this phosphate usually gets attached to another molecule or product. That is called phosphorylation, when a phosphate group is transferred, okay? ATP is a small molecule, and it's actually big and great to use for a quick burst of energy that can be harnessed to perform cellular work. It is very unstable, okay? So what happens, it's kind of like, think about it like if it's you and your 2 siblings together, okay? If you guys are forced to sit in a seat together side by side, okay, you guys usually are going to be repelling each other, which means unless you get along great, it's kind of like you're not going to have siblings that are going to be able to sit there for a long period of time and not try to push each other away. Okay, so that's how I like to think about it, is ATP. ATP is happy when it's just adenosine diphosphate. There's 2 of them together side by side, they're good. But when you add that 3rd triphosphate on there, it actually is too much energy withheld within their chemical bonds, so it makes them very unstable. And so that's why they're really great for a quick burst of energy, because it can easily be broken and then release that extra phosphate group to actually allow another molecule to be phosphorylated and so forth.